Good afternoon, everyone. So for this session, we have Dr. Sarita Das, who is currently working as an assistant professor in Barampur University. Before joining as assistant professor, he was working as a postdoctoral research fellow at University of Minnesota, USA, worked on the cloning and expression of recombinant bacterial antigens on virulent proteins and their use in disease diagnosis. She qualified CSI UGC NET JRF and Life Sciences in December 1990 and June 2000. She completed PhD degree in 2005 in biochemistry from University of Madras, worked on the molecular interaction between phytochemicals with pathogenic bacterial toxins. She has almost 20 years of research and teaching experiences. She has guided many MSc and MPhil students. Mrs. Das has around 32 research publications in referred national and international journals and has also published in several conference proceedings. So now I would like to request Mrs. Das to start his presentation.
Thank you. First of all, a very good afternoon to all the participants. And I must thank the organizers for giving me an opportunity to speak. Uh, so, so, is this slide visible?
Yes,
Am
ma'am.
I audible to
Yes,
all
ma'am.
of
It is
you?
visible. You are audible, ma'am.
Okay. Okay. Thank you so much. Thank you, Dr. Pratyus. Uh, I will be talking on the recombinant proteins as diagnostic markers. Actually, emergence and re-emergence of different types of novel and dreaded diseases had always been a threat, and their early diagnosis is required to address and combat the problem. And for that, we need so many different types of molecular techniques. And like other processes, diseases are also based on uh, the cause and effect the principle. Here, the pathogen or the antigen, which is of a protein or a glycoprotein of originated from the pathogen with an immunogenic activity, is considered to be the cause, and the antibodies or the immunoglobulins, they are also proteins produced by the host in response to antigens. So, for diagnosis, either antigen or antibody is measured, which requires a large amount of it, and biotechnology comes to play by producing a lot of it using a recombinant DNA technology. So I will be focusing on these aspects in my slides. So before going into the real topic, we need to know about the background and those major discoveries which opened the doors for molecular medicine. And to account that, the first discovery is on the transferability capacity of DNA. So this is actually done by Frederick Griffith in 1928. He conducted a set of experiments, and this Streptococcus pneumococcus, which causes pneumonia. There were two strains. One had a capsulated strain which was making smooth colonies, which is a virulent strain. So one strain was virulent S smooth colony forming capsulated one, and the other strain was rough, non-capsulated avirulent one. So when he injected the mouse with the smooth strain, the mouse died, and uh, smooth bacteria were isolated from the tissues of the dead mice. And when he injected the live R forms, avirulent forms, to the mice, he found that the mice was healthy, it was alive, and live R forms were isolated from the mouse. So that means pathogenicity is due to the capsulated smooth form. And then he used the heat killed S form, uh, and he found that the mice were alive and no bacteria was found. Then he mixed the heat killed S form with, means the dead S form bacteria along with the live R form bacteria, avirulent bacteria, and mixed them and injected to the mice. To his surprise, he found that the mouse was dead, and both the mixture of smooth form and R form were uh, isolated from the tissues of the dead organism. The mouse developed pneumonia and died. So that means something, since the dead bacteria is not capable of causing any disease, and the R form in such, like in its native naive stage, it is also not capable of causing the disease. So what change happened that made the mouse to be dead? That means something from the virulent strain is being transferred to the avirulent strain, because the avirulent R form is alive. It is capable of multiplying in the host, in the mice. So he was not able to identify what the material is. Instead, he left it as a transforming principle. There is something which is being transferred from the virulent S form to the avirulent R form and converting it to be virulent. So this mystery remained for many years, and it was not resolved what is the transforming principle till the discovery of three scientists in 1944, that is by Avery, MacLeod, and McCarthy. These three scientists, they used different sets of experiment and started using proteinases and ribonucleases and deoxyribonucleases, and they found that deoxyribonuclease use prevented this transformation, whereas the proteinases and ribonucleases didn't have any effect. That means that the transforming principle is a DNA. Okay, so the capacity of DNA to get transferred, that is one major breakthrough. Next, the double helix model that has been proposed by Watson and Crick and Rosalind Franklin in 1952, because unless until we know about the structure, we cannot work on its manipulation. So recombinant DNA technology, everything is based upon the manipulation of the gene. So uh, this is not possible unless until we know the structure. And the third step is the deciphering of the central dogma, which is actually discovered by Francis Crick. He proposed about this in 1958, and according to him, the double-stranded DNA helix is the repository of all genetic information that is stored in the cell. Means all the genetic content is being there with the DNA double helix, but the functions are being carried out by the proteins. So from DNA to protein, this how this information is being expressed in form of proteins that are called the gene expression, which accomplishes in two steps. First, from DNA to RNA. So DNA gets copied to RNA by transcription, and RNA being a mobile carrier, it takes the information from the DNA and then it comes to the cytoplasm where the ribosomes are present, the protein factories are present. These ribosomes encode the script and translate into the form of a protein. So this is the natural process which is occurring in all cells, but there are some deviations also, which we see with some RNA viruses or retroviruses, where in RNA viruses, RNA dependent RNA polymerase is used to copy RNA to RNA through RNA replication, and in retroviruses, RNA gets copied into complementary DNA by reverse transcriptase, and these unusual proteins are always targeted by the biomedical researchers. And coming to proteins, proteins are the nitrogenous, organic, large molecular weight most abundant compounds of the cell, made up of 20 amino acids, and they have diverse function, means starting from the structural units of membranes to the membrane transport proteins to different enzymes catalyzing mediates of biochemical reactions to the defensive proteins like antibodies and complements. So, such a diverse function of a protein is it comes from its structural organization. So, protein get organized uh, at different steps, starting from the normal sequence of a primary structure to a simple hydrogen bond formation in a secondary, then to some more hydrophobic interactions in a tertiary, to the cooperative action of a few proteins uh, combined together to function in its quaternary structure. Next, the recombinant proteins expressed from the recombinant DNA prepared through recombinant DNA technology. Again, in two steps. First, through gene cloning and then expressing the target gene in the specific biological system. It is useful in different biomedical research and disease treatment as drugs. And the first therapeutic recombinant protein is humulin, that is used for diabetes. So, almost 130, more than 130 recombinant proteins are already approved by the Food and Drug Administration of US, but still more than 170 or even more recombinant proteins are produced and used in medicine globally, because every day there is new discoveries and the number keeps on increasing. These are some of the examples of therapeutic recombinant proteins. So, just I'll brief on how important the therapeutic recombinant proteins, or how important it is to make recombinant proteins, because earlier, before the discovery of humulin, the diabetic patients were treated with the natural sources of insulin that has been taken from a cow or a pig pancreas, which had a little bit sequence difference, and that was not that effective, and also causing certain allergic reactions. Similar with the case of human growth hormone, which is used to treat the pituitary dwarfism. Just to treat a single person for 1 year from almost eight cadaver samples means after death, the brain samples were processed from there. These hormones are being purified and used. So using such sources is also not free of some what you can say uh, some risk. Yes, some risk, because sometimes through this transfer there is possibility of some disease transfer also. So usually the prion diseases called like the Creutzfeldt-Jakob disease, so they affect the brain tissue. So if the cadaver is having such prions, and when you take that sample and use to treat the dwarfism, the person may get infected with such kind of diseases. So it is quite risky to use the natural sources where this recombinant proteins comes to play, and they are much safer. This everybody knows about it, but still I'd like to tell you about the production of humulin or human insulin. Since the insulin gene is being isolated from the human cell, so human cell DNA that has been amplified for the human insulin is being used in cloning and then expressing the protein. So, since it is originated from the human DNA, that means it is completely safe because it is having the exact sequence of insulin. So this is a list of therapeutic recombinant proteins, and insulin, humulin actually in use since 1982, since 1985 human growth hormones, and some vaccines and interferons and interleukins are also used as the therapeutic recombinant proteins. So why do we need recombinant proteins? Because what we have in nature is very small, very scanty. But to meet with the demand of over increasing population and the diseases, we need more amount of recombinant protein, and that too in a pure form. So more and pure form we want. So this could be possible by using certain molecular techniques. So whenever we want to get some recombinant protein, so first of all, we have to amplify that gene, because the gene will be present in a very one or few copy number. So first of all, that specific gene has to be multiplied. Then only we can isolate that gene from the cell. Okay, so once that gene is been amplified and that is present in a large quantity, then we can isolate our specific gene of interest. Once the gene of interest is isolated and in a amplifier or in a much larger quantity, then we have to look for a suitable vector. Then we have to cleave both the vector and the target gene with the same restriction endonuclease, and then ligate it, and then put it into an expression system, which is usually uh, uh, the preferred system is E. coli, because that is the most easily means grown culturable and largely understood bacteria, I can say, because lot of work is been done. So this is the most user friendly, I can say, system to make our recombinant proteins. Then coming to disease diagnosis. So disease means dis-ease. So any suffering, like anything, if it is somewhat different from your normal condition, normal health condition, then it is considered to be a diseased condition. And according to the conventional methods, means earlier what how the disease were diagnosed, and now what is the difference in the diagnosis. So that I want to focus. So earlier the disease were usually detected upon the development of symptoms. So whenever a person had some symptoms and had some discomfort, then he or she visited the physician, and then they conducted a certain culturing of these samples like the serum, the blood, and the urine, sputum sample culturing, and then conducting certain biochemical tests. So that was taking more time. And then when symptoms appear, that means the person had already had a large amount of pathogen in his or her body. So when there are a large quantity of pathogen is present, and when it had already caused a significant amount of damage to the body, then only symptom comes. And that is, I guess, it's too late to uh, treat it. Usually, if there will be early diagnosis, means if anything will be uh, detected at a much early stage, uh, the treatment will be more effective, because our goal is to get effective treatment or get the person cured. Here two terminologies I will use: prognosis and diagnosis. Prognosis means detecting in advance whether a person is having any threat of developing a certain disease. So, and diagnosis, if a person is having a disease, then detecting. So, detecting after a disease, and that is diagnosis. And before disease, if you are assuming that this person is having a threat of developing a certain disease, then that is called prognosis. So, these recent practices use different molecular diagnostics for early diagnosis using different molecular probes, PCR, the real time PCR, ELISA, and others. So, probe is a short oligonucleotide sequence of almost 17 to 25 base pair, either radio labeled with P32 or non-radioactively labeled, like biotin or some other stuff. It is labeled but not radioactive. So, single stranded DNAs or RNA segments, and antibodies which could detect which could be detected through, if it is a DNA, then southern; if it is a RNA, then northern blood; or if it is antibody, then through western blood detection can be done. So PCR is used in case of suspected HIV, tuberculosis patient, or in case of mutation in case of cancer patient. Then ELISA is an immunological assay, enzyme-linked immunosorbent assay. It's used to measure either the antibody or antigen proteins or glycoproteins in biological samples like the blood, plasma, serum, sputum, urine, etc. And these ELISA tests are used for detection of pregnancy, HIV, hepatitis, or tuberculosis infection, etc. So this serological diagnosis or serodiagnosis for human infection is based on two strategies. First, uh, either on detection of antigen from the infectious agent, or the detection of antibody that is raised in the body of the host. So, but the second approach is detection of antibody is widely studied in diagnosis of many infectious diseases. Now, this slide shows a comparative account of the early diagnostic techniques like culturing in a plate and then conducting some biochemical reactions, and currently available techniques of using certain ELISA kits. There are so many different types of kits, are diagnostic kits are available, which is very accurate and also rapid, like diagnosis kit for this malaria or salmonella infection. So now, what is the characteristic of a good molecular diagnostic? It should be sensitive. That means it should be able to detect a very small amount of the target, and then it should be specific, means positive result from the target molecule only, and it should be simple, means it can run efficiently and inexpensively on a routine basis. And these are some of the protein based diagnostic methods. The list is here. Next, how does recombinant proteins are generated? If it is an antigen, then how it will be produced? So there is a sequence. First of all, to make any recombinant antigen, we need to know the sequence. So once we know the sequence, then according to its sequence, we can design the forward and reverse primer, and then we can amplify it, then isolate and purify and put it in the E. coli system, then grow it in a large scale, and then express the protein. And the protein could be purified using affinity chromatography. Once your protein antigen is available in pure form, now it is ready to be used. Next, the recombinant antibody production. So, for this, we need to immunize certain lab animals with the antigen against which we want to raise the antibody. So, first, the antigen is being administered to the lab animal, and then from its spleen, the B cells were isolated, and the positive B cells expressing the specific antibodies could be screened, and then the variable region of the antibody, that region can be cloned, and then large scale production of the antibody and its purification followed by its validation, means using its potency, or whether it is effectively working or not. First, whether it is available in the pure form. So that shows the X-ray film here. So through this we can identify it is available in the produced in the pure form, and then we can use it. Next, for recombinant vaccine production, we know that if it is an enveloped virus, then it will have some envelope proteins, or else the every virus has got the capsids. So either capsid proteins or the envelope proteins are can be produced in a large scale as a vaccine candidate. So here the hepatitis B virus, and from it HB antigen is antigen producing gene is being isolated, and then it was put into the recombinant E cell. Let me tell you, there are four different expression system. Till now, I had discussed about only one bacterial system, that is E. coli system, but still there are yeast cells, and insect cells and mammalian cells are also used for protein expression. because when we want to express certain eukaryotic protein which may have some introns and or some post-transcriptional modification, post-translational modification are also needed to come to the functional state, which is not possible in the prokaryotic cells since they don't have the facility. That's why we can use also the eukaryotic cells like the yeast cells or insect or mammalian cells. So this figure shows the sandwich ELISA once we get the antigen or the antibody. So if it is an antigen, then usually ELISA plates are 96 well microtiter pair plates, so which would be coated with the antigen and then with the primary antibody, then a secondary antibody which is specific against the primary antibody conjugated with some enzyme, and then you add the substrate. Then enzyme converts the substrate into a colored product which can be read in a ELISA plate reader. So either you can start coating your plate with the antigen or antibody. So if antibody is coated first, then antigen, and then the second antibody along conjugated with the enzyme. And similarly, substrate can be added and the color product can be the intensity or optical density OD value can be read using an ELISA plate reader. So this figure shows about the COVID-19 test kits. We are daily hearing about it, and it is based upon two principles. One is detecting the nucleic acid through the RT-PCR, and second one is the serological test that is available through the antibody test kits. So here the for anything I said in the beginning, like for working on any recombinant protein, we need to know the sequence first. So there's COVID, like the SARS-CoV-2 virus has been sequenced, and then the specific antigens are being produced, which are coated onto these handy strips. And whenever we want to do serological testing, then we can draw a small amount of blood or serum and then put it in the sample well, and it will move in through the capillary action and will show the result. Because there are two antibodies which are estimated, that is IgM, which is the largest antibody and developed first in the first week of infection, and IgG is the most specific one, and that develops later in the infection but last longer. And this type of kits are used for serodiagnosis, and in the first case the what you can say swabs, the nasal throat swabs are being taken, which is supposed to be contaminated with the viral nucleic acid, and then that will be put and run in the real-time PCR. Now the advantage of real-time PCR is that you can get the result in a much faster, means much early in comparison to the normal PCR, which after the completion of all the cycles only we can detect by running it in a agar gel. But here we can diagnose, means we can detect it much early. This figure shows the available HIV test kits. Uh, Next, coming to the subject like cloning, purification, expression of his tagged recombinant DNA is B, upon which I had worked. So I'll just what you can say, share my experience of making the recombinant DNA B. So there are several groups of Streptococcus. So Streptococcus is a very common pathogen, causing different types of diseases in human as well as in different animals. There are different groups. So group A, group B, C, G, so on, and they are named as GAS or GVS or GCS or GGS. But I had worked on the group A strep, uh, uh, and that is that GAS group. Streptococcus pyogenes is commonly called as the flesh-eating bacteria because they damage the soft tissue and cause necrotizing fasciitis. It is most common human pathogen and present in the respiratory tract of 5 to 15% of normal individuals without any sign of disease. Uh, they infect and invade the vulnerable tissue in immune compromised people. So if the immune status is good, then it doesn't affect much. But in case of immune compromised people only, it and also in young children, young school going children, they suffer from pharyngitis or tonsillitis, or together it is called as pharyngotonsillitis or strep throat, which can sometimes lead to life-threatening invasive disease like streptococcal toxic shock syndrome. And yeah, so this figure shows the different patterns of hemolysis by different groups of streptococcus. There are groups which completely, so this plate, the plate is actually of a SIP blood agar plate. So the RBC blood agar means RBC is present. The bacteria which is capable of completely degrading the RBC and the hemoglobin clears the red color around it, and they are called as the beta hemolytic, means the complete hemolyzers are the beta hemolytic groups, and streptococcus pyogenes is beta hemolytic. But streptococcus pneumoniae is alpha hemolytic, means it partially degrades the RBC and converts the hemoglobin into merohemoglobin, a green compound. So you can see a green coloration around the colonies. So that is alpha hemolytic and streptococcus pneumoniae. And other rest strains are gamma hemolytic, means they don't hemolyze at all. So no hemolysis are gamma hemolytic. So the Streptococcus pyogenes causes pharyngitis, and DNase B. There are actually four different types of DNases: A, B, C, and D. And DNase B is most common, and it play major role in GAS, means group A strep, that is Streptococcus pyogenes pathophysiology. Uh, uh, I want to tell you, like, if this common illness is been not addressed properly, then it can lead to some serious complications. Okay, so the antibody to DNase B is often used to confirm the clinical diagnosis of a previous group A strep infection, and the natural source. Like getting the DNase B from the natural sources requires complex and expensive growth media and handling loads of virulent bacteria. That is also challenging. So the present study is carried out to prepare his tagged recombinant DNase. So we can tag our protein with histidine, polyhistidine tags for its easy purification and easy detection. Okay, so all these things we'll see in the forthcoming slides. So, preparing the his tagged recombinant DNase in a non-pathogenic bacterial system, that is E. coli, For a commercial use, it can be it can be used in diagnostic test to detect the complications like rheumatic fever, rheumatic heart disease, which involves the permanent in some inflammatory diseases due to the strep infection, which permanently damages the heart tissue and heart valves, and also permanently damages the kidney. So it has got causing glomerulonephritis. So it has some nephropathic effect, some cardiopathic effect, and also it induces certain impulsive disorder in the patient suffering from this infection. Some neuropathic effect also it has, this group A strep has. So we need to diagnose this infection, which will be easier uh, for any lab to practice. So our objective is to obviously amplify the DNase B and to clone and to express and then to use it in the ELISA format and compare its result with the conventional assay. Means earlier the ADB means anti-DNase B titer in the patient were detected using uh, a what you can say, a chemical assay, I can say biochemical assay. I'll tell you about that a little later. So these are the different methods we followed. So first of all, we need to isolate the gene, that is DNase B gene, from the spiogenes group A stack, and we used 90226 strain, which has been isolated from the blood of a septic patient, and then its sequence is already known. So it has got a 816 base pair sequence. DNase B gene has got 1816 base pair. So according to the sequence, the primers were designed, the forward and the reverse primers. So we can tag with the histidine code either in the N terminus or the C terminus, but we used the C terminus to tag with the histidine codons. Then PCR amplification of the DNase B. Then the nucleotide sequencing is being done, because we have to check whether it had amplified correctly or not. So using the automatic sequencers, this was done, and then a blast analysis was carried out, and it found that our sequence had some 99% of homology with with the database with the other group A strap DNase B, whereas it didn't show any sequence homology with the other groups like group B, G, or C. Then the DNase B has been purified, and then the vector chosen is the PET28A vector, kanamycin resistant gene is the selected marker here. This both the target gene and the vector were digested with restriction endonucleases, then ligated, and transformation was carried out. Means once the recombinant vector is being generated, it has to be put into the biological system. So first, a subcloning efficient E. coli, that is DH5 alpha, is used, where cloning means multiplication of the recombinant vector is carried out, and then it was isolated and put it into the expression E. coli, that is BL21. So I used two strains, DH5 alpha, that is cloning efficient E. coli, and expression E. coli, that is BL21. So once it is put into the uh, expression system, means the BL21 E. coli that is grown in a large scale and then induced with the IPTG. Uh, Isopropyl galactopyrrolyside. So IPDG, because the T7 promoter is there. So upon induction, it produced the protein, the recombinant DNase protein. So once it is the production is over, means it is expressed within the BL21 system. Now, next step is the purification of the histagged protein. So for that, you need to what you can say, once it is there in the culture, so you need to centrifuge it, get your pellet, and then pellet is been what you can say, that is lysed with the lysozyme. So the E. coli cells break open and releases the protein, and then the protein is been passed through the nickel sepharate affinity column. So these columns and the technique is called immobilized metal affinity chromatography. So these uh, histagged proteins had greater affinity towards cations, metallic cations like the nickel, cobalt, or copper. So we can use such type of affinity column, and our recombinant protein binds to it. So we use a binding buffer. So binds it, then we wash the remainder, and then we use the elution buffer. So with the varying concentrations of imidazole from 50 to 500, we do first binding, then washing, which is not bound, and then eluting, the one which is bound, by disturbing the bond. So by disturbing the bond, we get our purified protein. So once it is uh, available, means 5 ml aliquots are being collected, means those buffers we will put in the top and then we'll collect from the bottom. So 5 ml aliquots are being collected, and each aliquot is tested for the presence of our, our recombinant protein in SGS page, sodium uracil. Uh, from every step, from starting from the wholesale lysate till the different illusion, from every step we have to collect the sample and store and run it in the SGS page to see its presence. And then the fractions which were tested positive, means those who had the presence of the specific DNase B protein, which is of 27 kilodalton. We'll see everything in the forthcoming slides. So I just narrated all the procedure. So this is how the primer is been designed. So the underlined sequence TCA, TGA, and the CTC and GAZ, they are the restriction sites. They were introduced into the histag recombinant primers, forward and reverse primers for BSP H1 and JO1, because our vector, that is PET 28A, has got restriction sites, similar isostigmeric similar sites of NCO1 and JO1. So NCO1 and JO1 is present in the vector, and here BSP H1 and JO1 is been the restriction sites for the recombinant histone. So we have to check with the positive control. So we had also used the original non-tagged primer, and this figure shows the first figure actually shows the PCR amplification of the DNase B. Line 1 shows the amplified DNA with the original. So that is 816 base pair, but the second line of first figure shows the recombinant hish-tagged DNHB that is somewhat heavier, I can say, of 840 base pair because of the addition of six extra histidine codons. So six extra histidine codons, the tag is used in the histagged primer. That's why the sequence is somewhat bigger of 840 base pair. And the second figure shows the double restriction digestion of recombinant factors. So before undergoing to the expression system, because you have to grow large amount of this expression bacteria, and you have to process everything, that itself is a I can say it's a, bit longer work. So before that, we need to check whether the recombination has taken place or not. If yes, then whether it is in correct orientation or not. Whether our target gene is oriented correctly or not. So for that, we conducted the double restriction digestion. So here the PET28A plus the histag radianase B, that is the recombinant vector, is been digested with BGL2 and the Jo1 because we ligated the BSPH1 and the NCO1. So BSPH1 and NCO1 in the front and actually messed up the restriction site. So we had to go 100 base pair upstream where a BGL2 restriction site is present and as usual the Jo1. So we used both the restriction endonucleases and cleaved the recombinant vector isolated from the DH5 alpha. I told you earlier that there are two strains we used. One is cloning efficient, that is DH5 alpha, and the BL21 expression E. coli. So the obviously M stands for the molecular weight markers, and in the second figure, lane one shows the restriction, double restriction digestion of DH5 alpha, and the 2 and 3 shows both the BL20, the recombinant vector isolated from the BL21. This is actually more sample is loaded here, 5 microliter, and here 10 microliter is added. So some extra bands of partially cleaved and uncleaved vector also it is showing up. But we got the target gene which is around 950 because of the extra 100 base pair upstream site of BGL21. So we are getting here a 9.5 kilo base pair target gene and the vector, fully cleaved vector of somewhat 4.9. So this proved that recombination has taken place and our gene is in correct orientation. So this figure actually shows depicts the nickel sapphirose or any metal, immobilized metal affinity chromatography. So you can see here in the bottom our recombinant protein or our protein of interest can get attached to the positive cations, and then when you use the elution buffer they get eluted and they can be collected and then they can be concentrated. So how we did the concentration? So I'll explain about this figure, but before that, so we can use certain tubes like this conical tubes with some cut-off value. So our sample is loaded with a 10 kilodalton cutup. So the proteins below 10 kilodalton, they will pass away with through the filter. Since our protein DNHB is 27 kilodalton, so it will be retained by the column. Okay, so this way we do the concentration. Once our illusion fractions, those who showed positive for the presence of histag recombinant DNase B, they were concentrated, and then we do a STS page, run the sample, and the arrow mark shows the 27 kilodalton DNase B protein. The one line and the three line, they are before concentration, and lane 2 and lane 4 is after concentration. So once we run it in the STS page, the protein gets separated. They are stained with the chromosobuline blue, and partly it is been transferred to the polyvinyl membrane by using wet Western blot. There are different types of Western blots: dry, semi dry, and wet blot. So we used wet blot and transferred it into the polyvinyl membrane, nitrocellulose membrane you can use. So once it is transferred, then the membrane is blocked with the 5% skim milk, and then it is added with the primary antibody, which is against the anti-His. So rabbit anti-His primary antibody is used and incubated and washed, and then second antibody, that is goat anti-rabbit secondary antibody is used, which was conjugated with the HRP, horse red peroxidase. Then a dab is used as the substrate and exposed to the X-ray film in the dark room and developed. So the lane 3 and 4 shows the X-ray photograph of the Western blot. X-ray photograph of the Western blot, lane 3 before concentration, lane 4 after concentration. So here we confirmed the presence of a histag protein. So using histag is very advantageous because you, for detection, for its elution, for purification, it's easier. We used the nickel cephalose column, and then using the anti-His primary antibody, which is easily commercially available, we can detect whether our recombinant protein is been produced or not. Detection is also easy. We don't have to go for the specific antibody against that particular protein. If it is a DNase B, then it could be only detected against the DNase B. But here, since the histag is there, so we can use the anti-His antibody. No need to go for the DNase B antibody. So, to use this DNase B as a gold standard, or to use it as a diagnostic marker, we have to rule out that this DNase B is present or is restricted to only group A strap, and it is absent in rest all groups of Streptococcus. That's why we used some 17 strains and conducted the PCR amplification against the DNase B, and then run it in the agar gel, and found that in rest all groups, starting from the lane 2 to lane 15, comprising of group B, C, and G. The DNase B was absent, but the positive strains in that is run in the lane 1 of 90226, and another strain uh, of group A. So both strains that is run in lane 1 and lane 16, they belong to group A step or Streptococcus pyogenes. So Streptococcus pyogenes had only the DNase B gene. In rest all strains, it is absent. So that means we can use this DNase B as a marker to diagnose the group A strep infection. Next, using our recombinant antigen, that is DNase B, in ELISA format. So, we had screened several serum samples which were collected from different countries. This work was conducted in University of Minnesota microbiology department. So samples from India, from China, and from US. These samples were for different studies, but they were used to estimate their anti-DNase B, means antibody to DNase B. So that is detected using the ELISA format. So you can see here uh, the first group of dots, that is of the DNase B label, means anti-DNase B in normal human. The second one is the rheumatic fever, and the third is the rheumatic heart disease patient. So we can see that the label of ADB, anti-DNase B label, is significantly high in the rheumatic fever and rheumatic heart disease patient. Next, the group B, colonized women. Chinese women serum, those who were colonized with the group B strap, and normal non-colonized women. So their antibodies were also estimated, and the first one shows the antibody label in the group B, colonized women. So we concluded from the PCR amplification assay that group this DNA B is completely absent in rest all groups except group A. Now, to our surprise, we found these group B colonized women had, means not all, but three outliers. You can see three women had very high level of DNase B, anti-DNase B, whereas the normal didn't have, means they had very low level, but these three women, they had high DNase, anti-DNase B or ADB. So we also assume that infection with one group makes the person vulnerable to further infection with other groups. So even though the person is infected with the group B, no doubt, since they have a high ADB level, that means they are also infected with the group A. So one infection facilitates the infection with the other strains. So that is what we predicted here. Next, this is from another study where the children, school going children from age 6 to 14 were enrolled, and up to 100 or more than that weeks they were screened, and their labels in different weeks for these antibody DNase B is been estimated, means the serial bleeds, we can say. So these patient has to visit the clinic, and their sample, serum sample, will be collected and stored, and if they have any infection or any step infection or any symptom, they will be like their swab will be cultured. So here we found that infection with the group A strap increased the ADB label, anti-DNase B label, significantly. Here we used two other parameters also, like the anti-ACPA. So ACPA is the C5a peptidase protein of Streptococcus. So Streptococcus C5a peptidase, that is also used because in response to the group A infection, group A strep has got different types of antigens. So just using one antigen won't be sufficient. So we used another antigen, like antibody against the other antigen, that is SCPA. So anti-ACPA and anti-DNase coincide. The effect after the group A infection coincide in both the cases, and here I will tell you conventionally how the tests were conducted. Means DNase B antibody, how it was measured conventionally means earlier. I spoke about the DMEM method, that is DNA methyl green micro method. DNA methyl green micro method. DM DNA methyl DMGM. Okay, so here the DNA has got affinity for a dye that is methyl green. So methyl green will bind to DNA and will form a green color. So that green color when will be added with the DNase. Okay, so DNase will break. DNases are the nucleases which has the capacity to break DNA. Okay, so these they will break, and the color will fade up depending upon the concentration of a DNA. It will fade up. So when we add the antibody, it will interfere. So inhibition of the neutralization assay, that is measured in the micrometer. That is used in the conventional practice. Means earlier the DNases, the antigens were extracted from the culture filtrate of Streptococcus pyogenes. Means the Streptococcus pyogenes will be grown in large quantity, and its toxins, its means the secretory proteins will be released to the culture filtrate, and from there it will be collected. But now, so you have to handle so much of high concentration of virulent bacteria, and also the process is very tedious and it is less. But using recombinant DNA technology, we can get the recombinant antigen in a much easier way without having any contamination. Because when you go for the culture filtrate collection, that your protein of interest will be also contaminated with other such streptococcal proteins, which will interfere with your experiment. But in recombinant proteins, this is omitted. Means it is means the only that gene is present, so that product is there. So there is no contamination from the streptococcus. But in the diagram we saw some extra bands, but that is from some histidine rich E. coli proteins. Okay, so E. coli proteins are not going to interfere with our result. So, this is another set of experiment that has been carried out in the participant. The previous one is Alabama 14, and here it is 768 participant number. This patient is a 8-year-old boy, and he had 3 episodes of group A staph infection with the serotypes M6, M1, and M49. And upon exposure, means upon the infection with the group A, this antibody label raised, which coincides with the ACPA ELISA results. But the again with in due course it falls. So actually whenever there will be any active infection, the antibody level will rise, and depending on the half life of the antibody, for uh, this ADB it is around 48 weeks, and for ACP it is around 55 weeks. So automatically after that many days it will decline. The label will decline. So, which is clearly detected in the ELISA assays of both anti-DNase B, ADB ELISA, and the anti-SCPA. But the conventional method, the second picture is comparing with the two anti-DNase B estimated through ELISA and through the conventional method. So the conventional micro method, where we can see it is much flat. We can't see the differences, the week-wise down and fall with exposure to the group A infection, how it rises and how it falls. So it is not clearly demarcating that it is somewhat stiff, but in case of ELISA, it is much more sensitive to the rise and fall in ADB labels. Next, uh, coming to the discussion, so using RDT, sufficient amount of desired proteins of deadly pathogens can be expressed in an efficient non-pathogenic system like the E. coli without handling loads of virulent bacteria like the Streptococcus pyogenes, that is a virulent pathogenic form. So we don't have to handle them. Simply just a very maybe 3 ml of culture we can do and isolate the genomic DNA and amplify our special gene of interest, and then go for its PCR amplification, then purify it and cut it and put it into the vector and make your recombinant vector and express it. That's much easier than culturing loads of virulent pathogenic forms and collecting the proteins from the culture, and which is often contaminated with other streptococcal proteins. DNA sequence with a string of 6 to 9 histidine residue is frequently used in vectors for production of recombinant proteins, which can be purified and detected easily as it binds to the immobilized metal ions, and the process is called the immobilized metal affinity chromatography, so IMAC. His tagged proteins can be easily detected by commercially available anti-His antibodies, and DNase B is not detected in the PCR or DNA samples from the rest all groups except the group A step. So this could be used as a diagnostic marker. Anti-DNase B-based ELISA proved to be very sensitive and responsive to changes. We saw that in the two sequential serial bleed samples of two patients, which is much more responsive to changes in comparison to the conventional micro method. And use of recombinant DNase B also eliminates the variability associated with the enzyme, and purified streptococcal supernatant cultures from various commercial sources. Okay, so this also confirms that ELISA assays of anti-SAPA and ADB, anti-DNase B, would be useful indicators of recent group A strep infection. So, to conclude, there is a pressing need for development of rapid, easy, and accurate test kits to diagnose different diseases or detection of various conditions, which could be resolved by using recombinant proteins. Recent resurgence of deadly group streptococcal infections and their sequelae urged for an accurate clinical diagnosis and laboratory confirmation of group A staph infection. Therefore, his recombinant DNase B was developed and that may be used commercially for group A step detection and to study its sequelae. So these are some of my references, and this is the publication from my work. Thank you.
Thank you very much, ma'am.
Thank you very much, ma'am.
Thank you. If you have any queries, I would love to answer.
Any participants, those who are having any queries? Are there any queries, participants?
I usually tell my students like if there is no queries means either everything is understood or nothing is
They haven't
understood.
understood anything. Yeah, yes, right.
So if there is any query, then you can ask me.
Or else, I would also request the participants to please forward the questions to me so that I can send it over to ma'am to get it answered, and we'll get back to you later on. Okay, thank you, ma'am. So that was a very nice presentation. And ma'am, we also
Thank you.
expect your presence at evening time at around 5:30 for the valedictory.